Keys found in here on Monday. All right, so we do have an exam Monday. All right, so homework due Monday. That chapter 7 homework is a little bit longer. There's more animations with it. Again, don't wait till the last second. You can always look at that homework again. So, you know, try to not make that such a burden. Try to get on that ahead of time as much as you can. I did post some practice questions. There's a tab on Blackboard. It says exam objectives and practice questions. So I think like 12 questions and answers. So look at those sometime. I did give the last exam to just a handful of you online last time, just because of quarantines and all that stuff. I'm not planning on doing that this time. If you are sick, by all means, we can do a makeup. We'll try to schedule that in a few days after Monday. So, you know, whatever, something comes up, just be in contact with me. But I'm not planning on doing it online this time. So, all right. So when we left off last time, and we just got a little bit to go here that will still be included on Monday's exam, and then we're gonna kind of do stuff that won't be on it. I felt like I kind of hurried through this at the end. Last lecture, we talked through translation and regulation, talked about different kinds of mutations and the effects that they have. And we'll get to a couple practice questions in a second. But when we finished up, we were talking about different kinds of horizontal transfer. So this is other ways that a cell's genome can be altered. And the first of those ways was transformation. So that's when a cell will take up DNA from its environment, usually released by some dead cell where the cell wall has broken down. So the experiment that proved that can happen was done by Frederick Griffith in 1928. Okay, so this was well before we knew the structure of DNA, well before Michelson and Stahl's experiment that talked about how DNA is replicated. So kind of interesting, but remember, he took a living strain of Strep pneumoniae that wouldn't cause disease and mixed that with a dead strain that would. So those dead cells, that the DNA that at least encoded for the capsule production anyway, was taken up by that living strain, and the mouse died. So again, that's the evidence for transformation. Okay, the second kind of horizontal gene transfer is transduction. And if we had talked about viruses already, then this would look a little bit more familiar. Just know that this involves a virus. And we're going to talk more about viral replication, but what you need to know basically here to understand this is that there are viruses that only infect bacteria. We call them bacteriophage. Phage means to eat, so bacteria eaters. And some of the bacteriophage look really kind of bizarre. They've got these weird spindles and tail fibers on them. But in a nutshell, a bacteriophage can land on a bacteria cell as part of their replication. When they inject their DNA, they're going to break up the host DNA. And then some of that host DNA could accidentally get packaged in with the phage DNA when it lyses out of the cell. And then when it goes to another cell, lands on another cell, if it does a different kind of replication that we'll talk about later, it could actually integrate some of that DNA from the old cell here into the new one. Okay, so it could actually change the phenotype perhaps of that new bacterial cell. right? So we can kind of do this on purpose in the lab in kind of a either generalized or specialized way, kind of pick out certain genes on purpose, but that can potentially happen in nature as well. And then the last way of horizontal transfer is called conjugation. So this, we've mentioned this a few times before. With bacterial conjugation, the cell produces a conjugation pilus. We talked about that structure, fimbriae and pili. Conjugation pilus brings the cells close together. Okay. A pore opens up. You'll see that better on the next slide. And then the DNA can be transferred. So we thought originally that the DNA transferred through this pilus. I don't think that's the case. It's just a mechanism to bring the cells closer together. Both cells are alive. Okay, so with the other two mechanisms, you know, one of the cell that was giving up the DNA was dead. So that's another difference. Oh, and we call the donor cell F+. Okay, so that means it has fertility plasmid. If you remember back a couple of lectures, we were talking about different kinds of plasmids, like a bacterial resistance plasmid or bacteriocin plasmid. A fertility plasmid enables an organism to make that pilus and extend that to a F- cell, one that doesn't have the fertility plasmid. So the question that came to my mind is, well, how does an F+ cell know that that other cell is F-? It probably doesn't. It would just reach out to any cell. It would not be able to attach to a cell that was F+. Okay, so an F+ cell, they make some proteins on the surface that makes it so the pilus can't attach. Okay, let's see. So if you kind of look at what's going on inside of the cell during conjugation, here's the donor cell here on the left. It has that F plasmid. It's made a pilus to bring the cells close together. A pore opens up here, and one strand of that F plasmid is transferred into the other cell, and then the complementary DNA strand is just made after that. Okay, so now this cell on the right is an F+ cell. So the so what here is that the F+ cells often are not just transferring their fertility plasmid. This can be a mechanism to transfer other plasmids as well, like a antibiotic resistance plasmid. All right, so we think that this is one of the more common mechanisms that a cell might share its ability to survive, you know, in the presence of some antibiotic. Okay, we know that some cells are a little bit more promiscuous, you could say, that are doing this a lot. So they're sharing this with lots of other cells. It can be a cell of the same species. It can be a cell of a different species. So we think that's how other plasmids get shared. Your book goes into a little bit more detail there and talks about how sometimes that F plasmid will actually incorporate into the cell's chromosome, and then we call it a high frequency recombination cell. We're not gonna worry about that, but there is kind of a little step further you can go there if you're interested in reading about that. Okay. Questions. That wraps up chapter 7. We are actually ahead of schedule. If you look at our syllabus, we've somehow gotten ahead. No worries. I have plenty of stuff to talk to you about, especially as we kind of crunch up more at the end of the semester. So no worries about that. But let's take time and do a couple practice questions here. Okay. So really here, this is kind of asking, do you remember the terminology involved with these different mutations? So I'll give you a minute. You can discuss it with each other if you like. All right, let's see what you think, and this would be more fun, I know, with the clicker question. So remember, if you have a silent mutation, that doesn't make any difference in the amino acid sequence that's made. So you can cross that out. That's not going to make any difference. Now, typically, remember, a nonsense mutation is going to encode for a stop codon. So typically, that's going to be really bad because the whole rest of that protein wouldn't get made. But here I said it's at the very end of the messenger RNA, so that actually may not have too much of an effect. Remember, a missense, and here in the last 6 of a sequence, that doesn't really matter where it is. Missense just means it codes for a different amino acid. Again, I said that might make the protein not functional. It might not have any effect at all. But if you have a frame shift in the 5th codon, so that's right at the beginning of the transcript there, then that all the rest, everything after that is going to be wrong. Okay, so likely number 3 is going to have the biggest effect, just because, yeah, you know, all the amino acids after the very beginning are going to be different than what they're supposed to be. So, all right, so again, you want to know the difference between nonsense, missense, and silent. Okay, which can be confusing. And I came across this. I know it's kind of hard to see it. It's not very clear. But a diagram like this, I thought, oh, there's all sorts of things that I could ask you about this. So starting kind of from the easiest and then getting harder. What process is this picture talking about? Okay. Replication, right? We got double-stranded DNA and it's being copied. Okay, then I could ask you, well, what are these different enzymes? So enzyme A, okay, that's looks like it's unzipping the 2 strands. That's helicase. If it was up further here, if it was up here, what's the enzyme that works upstream of the replication fork that relaxes the supercoils? Right, that's gyrase. Good. And then enzyme B, that's the main enzyme that's doing the replicating. That's DNA polymerase 3. 3 there, and 1 on. This is the leading strand on the bottom, and up here, actually this time this is the lagging strand. And maybe then also to think about the directionality. So I could say, well, what end is this up here? Okay, is this the 5' end or the 3' end? Right. So work your way back. Right here, this is going to be the 3' end because it's adding on to the 3' end. And so, you know, it's anti-parallel. So that would be 5' up there. Okay, so on the bottom here, this is 3'. Wait, did I do that wrong? 3' and Hmm. Oh, wait. Did I do that right? Okay, so then. Oh, right, yes, I did that right. Okay. See, I mix myself up sometimes. Yep, because this would be 5 prime. Yep. So then their strands are together. Yep. So yeah, I like to ask that question or have you think about that question because it's always goose me up sometimes too. Oh, one other enzyme over here, this enzyme C, kind of after the process is almost done, right? That's going to be ligase to kind of just seal up the, add some bonding in there. Remember, the primers have to be taken out. DNA polymerase 1 fills in where the primers were. RNA primers were temporary, and then the ligase seals it up. So, okay, little review there. Okay, any other questions? Okay, so that's the end of the material that will be tested on. Let's move forward. Talk a little bit about viruses. Okay. A topic that hopefully all of you are interested in, because we have to think about it every day. So we're going to start out just thinking about some basic characteristics of viruses. What kind of structures do they have? How do we classify them a little bit and think about their replication as well? There's some differences, like I mentioned earlier, there are viruses that only infect bacteria. We call those bacteriophage. How are those different than viruses that infect animals? We as humans, I would consider an animal, and we'll finish up our discussion, I assume next time, into prions, and just talk briefly about some prion disease, kind of some scary stuff. All right, so just by way of an introduction, I like to point out, when we first started meeting, that bacteria are not all bad. We spent an awful long time talking about bacterial infections, but, we couldn't be healthy unless we had bacteria in our bodies. For viruses, it's a little bit different. Like somebody asked you, do you want a virus? No, thanks. I can't see that that's going to benefit me in any way. And we can have a lot of interesting discussions, I'm sure, talking about which viruses you have encountered and which ones I have encountered. There's a lot of them that will make us really sick, and we're often not very good at treating viral infections. We kind of have less tools to work with because they infect our cells. They're inside our cells, so how we get to them without hurting ourselves. I recently came across an article that's a few years old now, but can viruses benefit humans? And this whole article was trying to show ways that they actually could. A couple of the things that they mentioned in there is that there is some evidence that there may be some symbiotic relationships between some viruses and some cells. But the real strength was in potential medical applications in as much as that we could actually manipulate viruses a little bit and use them, things like to target cancer cells, for instance, to get our immune system to target the cancer cell by infecting tumors, say with viruses, or as just a delivery mechanism. Okay, some of our vaccines, our RNA, our DNA vaccines use a virus to get those pieces of nucleic acid into our cells. So already reaping the benefits of viruses in that way. So there are some potential medical applications that are that we can thank viruses for, but sometimes it's a little bit of a stretch. And I always like to show this slide. So 2 years ago, February 5th, 2020, I showed this slide and I said, hey, this is really interesting. There's this virus in China that's killed 400 people. Oh, this is kind of interesting to keep our eye on. Not even thinking that it could, changed everybody's lives the way it has. So at that time, we were calling it the novel coronavirus 2019. So I always like to do a case count every time I have taught this class. So roughly every 6 months or so, the case count has doubled. So even since the end of September, both worldwide cases and US cases has doubled, which is kind of mind-boggling. I think we're not as good now as keeping track of cases now that we have a lot of at-home tests and people aren't reporting when they are positive. But so cases have continued to skyrocket. Thankfully, the deaths are not doubling every 6 months. I think we have about 200,000 more deaths now than what we did at the end of September. So, but we're closing in on 1 million deaths. So that's pretty sobering. So we will talk more about coronavirus. I like to show you just some examples and talk about examples of viruses that you've heard of, but right now we're just thinking about kind of characteristics of viruses. Later on in the semester, we'll pick out some specific ones, and that's when I want you to know, oh yeah, coronavirus is a single-stranded RNA virus, for example. Right now, just lots of examples I'll throw at you. A few years ago, the one that was always in the news was Ebola virus. And that was the one that we were always talking about. I think there was around like 7,000 deaths, like 2014, 2013 epidemic. But this slide was meant to point out, hey, viruses can be life-threatening or they can be really super mild. So here's a picture that I've used for a long time of coronavirus, meaning coronavirus can just, give you a little cold. It's a common cause of a common cold. And then viruses that infect bacteria can look pretty strange. They've got this head and this tail, these tail fibers. It looks more like a little robot almost, or a little lunar lander. Not all bacteriophages look like that, but some of them do. Thinking about size, first of all, they're really small. We only could visualize viruses when we had the ability to use electron microscopy in the 1930s. We knew that they were around before that, some infectious agent that was really small, we just couldn't visualize it. So if we kind of compare how small they are, here's a red blood cell compared to an E. coli. And then you blow that up further, the E. coli compared to some different viruses. They vary in how big they are. The biggest virus, I believe, is the smallpox virus. Some viruses are so small that they're more close, the same size as a bacterial ribosome. So there's some variation there, but in general, way smaller even than a bacterial cell. Okay. So let's think about what viruses are made of. They are acellular, so that means they don't have the same structure as a cell. And in fact, we'll talk about later, do you consider them living? Most scientists would say no, but there is room to kind of disagree on that a little bit. All viruses are going to have some kind of nucleic acid. Okay, so they're either going to have DNA or RNA. They're not going to have both. Okay, they have one or the other. The capsid, okay, that's this protective coat that we find around the nucleic acid. Okay, so we'll call this, it's made out of protein, a protein shell to protect either the DNA or RNA. So all these little, in this picture, you can see all these little subunits. We would correctly call those capsomeres. Okay, again, they're just made out of protein. And if there is nothing else around that, like a membrane, then you would have the virus's kind of mechanism to attach to its target host cells there's going to be proteins in there that are going to help it attach to whatever kind of cell it needs to attach to. Okay, but those attachment proteins are always going to be on the outside. So some viruses then are going to have an envelope around the capsid. And that's just going to be a phospholipid membrane. Okay, so that makes sense, the composition of it, because viruses are just basically stealing that membrane from the host. It can be actually from the nuclear membrane, more often from the host cell's plasma membrane. Okay, so that's what the composition is going to be. And a couple of terms that we'll try to use correctly. When a virus is outside of a cell, we would most correctly call it a virion. Okay, so that means it's got the nucleic acid and the capsid, and then the envelope if it's an envelope virus. Once that virion is inside a host cell, the membrane and the capsid are going to fall away, and just what's left is the genetic material. Okay, and then we would correctly call it a virus. Those two terms aren't always used correctly, and we will try to do that. Okay, often we just say virus kind of for everything, but anyway, we'll try to use them correctly. All right, so if we kind of look at, well, what do viruses look like? There's different terms that we can use to describe their shape. And most viruses, all viruses are going to fall into one of these 5 categories. Helical, meaning the capsid is considered helical. We'll look at some examples in a minute. Here's an example of an enveloped helical virus, and that just happens to be the SARS virus, SARS-CoV-1, severe acute respiratory syndrome. And then there are capsids that are considered polyhedral, enveloped polyhedral, and then there's a class of viruses that just don't really have any kind of symmetry, and they're just kind of the miscellaneous ones, and we'll call those complex. All right, so if you look at a capsid with helical symmetry, it's just kind of got this long tube that covers and protects that nucleic acid, whether it's RNA or DNA. So an example of that, tobacco mosaic virus, the virions just look like kind of these long sticks. But that helical symmetry kind of gets covered up if the virus has a membrane around it. So then it just looks, kind of more roundish, I guess, like the influenza virus. There are viruses with that polyhedral symmetry. Okay, and I think I couldn't find any examples of polyhedral symmetry that wasn't actually an icosahedral capsid. Okay, so an icosahedron, that's a polygon that has 20 sides. Okay, 20-sided or 20 sides. Okay, so it's got 20 equilateral triangles for each face. Okay, so this kind of symmetry, icosahedral symmetry, it's just very symmetric because of all those equilateral triangles. And so it gives the virion high strength and stability. Okay, so that's probably why it's so popular. It's a very stable, very strong structure. If we looked at this a little bit more closely and thought about the kinds of proteins that make up the capsid, we would call the ones in the corners, well, that's so here, the corner ones, penton proteins. Okay, so these are the corner capsomeres. an icosahedron is always going to have the same number of corners. It's always going to have 12. So in this picture over here, the pentons are in blue. Okay. And then the hexon proteins are the ones on the face or the sides. And the number of hexon proteins could be quite variable. It depends on how big the virion is. So for example, this example on the right here, this is an adenovirus. It's named adenovirus because it was isolated from human adenoids. Makes sense. But here there's 12 pentons and 240 hexon monomers. Okay, so the more hexons, the bigger the virion would be. Okay, and if there could be you have a icosahedral capsid, and then it's covered in a membrane. Herpes virus is an example of that. Don't worry about this tegument proteins. We're going to talk about that later. And then the last category, complex. Again, these are virion shapes that just don't fall into any particular type of symmetry. Smallpox, that's this one on the right here, that's always considered complex. It's got this really kind of bizarre shaped capsid. Oftentimes rabies up here is considered complex. It has a membrane that, because of the proteins that are in it, it gives it kind of a bullet shape, which is kind of unusual. And sometimes we consider it bacteriophage complex just because it's got this spindle and tail fibers. And you can see here that the head is elongated just a little bit. So those aren't absolutely equilateral triangles there, but it's a little bit variation of a icosahedron. Okay. All right, let's look a little bit closer at the viral envelope. So what kind of viruses have envelopes? This is going to be most common for viruses that infect animals. And in fact, there's only one bacteria phage that I know of that makes an envelope. All the rest of them are naked or non-envelope. So really common for viruses that infect humans to have an envelope. Not all of them, but that's pretty common. And like I said, they're acquired from the host. I talked about that already. Phospholipid bilayer. It may have these glycoprotein spikes that are going to protrude out from the envelope. Of course, we know all about spike proteins from coronavirus. A lot of attention is made to those spike proteins because that's the parts of the virus that our immune system recognizes the best. So all of the vaccines, it's all been all about those spike proteins. The spike proteins for influenza, we're going to talk about these more later in the semester when we talk about the flu, but these hemagglutinin and neuraminidase. So H and N, you've heard of H1N1 flu, for example. So there's different variations of those spike proteins that give us kind of different strains of the flu. Now, some naked viruses can have spikes as well, as we saw with adenovirus. And not every virion will have those spikes, but the spikes are really important to aid in host cell recognition. If a virion doesn't have spikes, then it will have some kind of proteins in its outer layer that will aid it and help it recognize the specific kind of cell that it's able to infect. Okay, sometimes the spikes will help them with entry as well. One other interesting thing is typically enveloped viruses are pretty wimpy in the environment. So let's say envelope viruses, we'll say they degrade quickly in the environment. And you might think that it would be the opposite, but that phospholipid membrane, it just dries out really quickly. So I'm talking about in the environment, I mean, maybe on your desktop or whatever, or on the computer keyboard. That phospholipid membrane dries out really quickly. It's really susceptible to, you know, sunlight, any kind of radiation, or any kind of disinfectant that we put on it. So envelope viruses, like coronavirus, typically may only survive an hour or two, or be viable for an hour or two on some surface, maybe up to a day or so. So when they were first saying that coronavirus, you know, leave your groceries in the garage because, you know, somebody might have touched them that had COVID, we think that's not necessary anymore, because, you know, there's exceptions to every rule, but in general, those envelope viruses don't survive well in the environment. Now, inside the host, it may give the host an advantage and help, you know, it evade our host immune system. Naked viruses, on the other hand, survive for a long time on surfaces, and they're a lot harder to kill with disinfectants. So something like norovirus, they even say that hand cleaner doesn't reliably kill norovirus. All right, that's the virus that makes you like come out at both ends. It's just miserable. That can survive for even up to weeks on surfaces. So looking at the structure of the virus can kind of help you understand a little bit about how hard it is to kill. And because of those viral spikes or surface proteins, viruses are often very host specific. So here's just some examples. Here's an example of a tobacco plant that's infected with tobacco mosaic virus. And actually, just look this up. That particular virus actually infects a whole family of plants that includes like tomatoes and petunias, which I was not aware of. But anyway, a tobacco mosaic virus is not going to harm us, right? They're specific for a certain kind of plant. Bacteriophage, here's a bacteria cell that's in trouble. There's a lot of bacteriophage surrounding it. But bacteriophage typically will only infect a certain species of bacteria. So we have phage that only infect E. coli or only infect Pseudomonas. So there's another way that we may be able to use viruses enable us to kill bacterial cells and help us conquer bacterial infections. In this picture here, these little pink dots, those are HIV particles that it looks like are attached to some kind of white blood cell, maybe a helper T cell. HIV, right? We can only be infected by certain ways, like through blood contact, through sexual intercourse. If you swallow HIV or you breathe it in, you're not going to get sick. It's a certain kind of receptor it needs on certain kinds of cells. Very often viruses will only infect a certain host. Now, certainly we've seen with coronavirus that, the tiger at the zoo got coronavirus, got COVID, or pets, you've heard about pets getting COVID. So it appears that coronavirus is a little bit more of a generalist. West Nile virus is another one that will infect lots of different species. But that's not as common. Oftentimes they're very specific. All right, so zeroing in a little bit further on the genetic material. So what kind of nucleic acid? We said either RNA or DNA. Okay, so it could be single-stranded DNA or double-stranded DNA. we could have single-stranded RNA or double-stranded RNA. Okay, so there's two kinds of nucleic acid that you don't see in any other life. You never see single-stranded DNA and you never see double-stranded RNA. All right, so that when this was, different viruses that had those kinds of nucleic acid were discovered, that was really a mind-blowing thing. And there's actually, we'll talk about this in a minute, you can have plus sense single-stranded RNA or negative sense. We'll talk about the difference between that in a minute. And in addition, that nucleic acid can either be in a circular form or it can be linear. And if it's linear, it could be either segmented or unsegmented. We'll say non-segmented. Okay, so for example, this virion over here, that's got a segmented genome. Okay, you can see three different pairs of lines there, whatever that is, RNA, I think, most likely segments. Coronavirus is unsegmented. There's just one long string of RNA in that genome. Okay, so little variety there. What do we know about the number of genes? It's pretty small. Okay, so how small is it? In humans, we know that there's somewhere between 25,000 and 40,000 genes. Before the Human Genome Project, we thought it was more like 100,000 or 200,000. So that was kind of a surprise. We can't say exactly because just from knowing the sequence, we can't know exactly what makes a gene up, but genome. Compare a typical bacteria to humans, it's about an order of magnitude less. So about 4,000 genes for E. coli. Viruses now are going to be even much more than that. The virus with the smallest genome, hepatitis B, only has 4 genes. Now, other ones like herpes virus have somewhere between 100 and 200. And the analogy that I'm very proud of, that I've come up with this analogy, I know a couple of you have heard this already, but I like to call my son a hepatitis B virus. And sometimes he asks me, why do you call me that again? So bear with me. So this is a picture of my son. This is one of my most favorite pictures of my son. I don't know why I'm so proud of it. He's actually the B, and that's not why I call him hepatitis B. But anyway, so this is a mental picture of my son. So this was at the Washington game last fall. So, and they actually used house paint, latex house paint. I mean, it looks good, but that couldn't have been easy to get off. I know they forgot a couple times, but overall, I gotta say they did a pretty good job. So this is why I call him hepatitis B virus. So a couple of years before this, he came home on fall break. So fall break, 4 days at home. He hitched a ride with his friend Micah here, he's the U, and I went and picked him up in Grand Rapids. And he hopped into the car and I said, well, wait a minute, where's all your stuff? He had with him his laptop and a football to play catch with. That's all he brought home for 4 days at home. I had a little mom sigh, really, but the more I thought about it, okay, he's coming home. He's got a bed to use. He's got sheets. He's got towels. He's got food. He's got old clothes he can wear. He's got an old toothbrush. He's going to take over our house and use everything in our house. He didn't really need to bring anything with him. Okay, so hepatitis B viruses, they're very efficient at taking over a host cell. There's only a couple of special things that they need to bring with them in order to reproduce. Okay, so that's why I call my son hepatitis B virus, because he travels light, right? He just kind of uses whatever is available. So what kind of, and so here's just, we have ways to break open cells and to visualize the DNA. So here's a partial genome of E. coli, and this little bit right here is some bacteriophage that has infected E. coli. So, you know, really just a very small number of kinds of genes that a virus needs to bring with it. So the question is, what kind of genes is necessary? What do viruses information, what kind of information do they need to bring with them in order to be successful at reproducing? Well, here's just some examples. They may need some information that would encode for viral spikes. They might need to regulate the host cell in some way, turn certain genes off, turn other ones on to help them replicate themselves. Usually a big one here is replicate. They have to have a way to replicate their genetic material. So if they're a DNA virus, okay, and so in the past, sometimes I've taught this before I talked about genetics, you know, they just take over the host machinery. They've got, you know, the enzymes to replicate the DNA, to transcribe the RNA and translate into proteins. But maybe you've got an RNA virus. So if you've got an RNA virus, they're going to need to have some way to replicate their RNA, right? We never do that. We never make RNA from RNA, right? We always make RNA from DNA. So they may need some kind of RNA dependent polymerase, RNA dependent RNA polymerase. Okay, so sometimes you see that RDRP. Again, so that's an RNA dependent RNA polymerase. So our RNA polymerase uses DNA as a template, right? So they may need to have, sometimes that's referred to as replicase, an RNA dependent RNA polymerase. Some viruses, some RNA viruses will use an enzyme called reverse transcriptase to make DNA from RNA, and then to make more RNA from that DNA template. Okay, HIV is famous for doing that. Okay, so again, in our cells, we never make DNA from RNA. So if that's a virus's strategy, they got to bring their own enzyme or the information to make that enzyme themselves. All right. So just kind of finishing up kind of our, you know, basic things about viruses before we talk about how they're replicated. We have a couple of different ways that we will classify viruses. And one way we refer to as this hierarchical classification. And the other way is called the Baltimore Classification. David Baltimore was one of the people that is given credit for discovering reverse transcriptase back in the 70s. So he made his own viral classification and it kind of stuck. So with this hierarchical classification, basically, we've tried to treat viruses like they were living organisms and put them in that same classification system, right? That doesn't really always work out so well. But with this system, there's 3 orders classified. So it goes phylum, class, order. I don't think there's really classes or phylums of viruses, but there's 3 orders. And then below that is the family. So all viruses are classified in families. And we know it's a family name because it's going to end in viridae. So herpes viridae, that's a big family of viruses. And if we're going to be grammatically correct, we would italicize family names. The genus names will always carry the suffix virus. Right, so herpes virus is in the family herpes viridae. And again, it's correct to italicize that. How do we put viruses into these genuses and families? Well, we look at shared characteristics, things that we can see for the most part. So what's their nucleic acid? Is it DNA? Is it RNA? What does the capsid look like? Is it helical? Is it polyhedral? Is it icosahedral? Is there an envelope? To some extent, just kind of the shape and how big the capsid is. So those are the kind of the four main things that we use to put viruses in one of those groups. Here's another example, and here I've included the species. So a particular virus, it's in the family Bunyaviridae, the genus Lebovirus, and then the species, I'll write this in, I didn't add this here. The species is just the name of the disease that is caused. Okay, so in this example, the species, it's Rift Valley fever. That's the disease that this virus causes. And then you tack the word virus on the end of it. So Rift Valley fever virus, that's the description of the infection that we suffer from it. Okay, so sometimes, you know, a lot of times it's not this complicated. Like I said, for herpes virus, okay, herpes virus, it causes herpes in us, and it's in the genus. Herpes virus is also the genus, really. Herpes viridae is the family, so sometimes it's a little bit more intuitive there. There is this, you know, and of course with this classification, because we're looking at characteristics that, you know, we can argue about what's more important, and, you know, we're always kind of refining our classification system. So some of that's kind of, you know, it changes and as we get new information. With the Baltimore system, it's a little bit more cut and dried. All viruses are put in one of 7 groups. And don't memorize which is which here, but it's all based on nucleic acid. Okay, so for example, double-stranded DNA virus, that's class 1. Single-stranded DNA, class 2, for example. Then there are, you know, plus sense and minus sense retroviruses they're single-stranded RNA plus sense, but they go through a DNA intermediate to make more RNA. And this last group, this group includes hepatitis B virus. It's kind of funny. It's a double-stranded DNA virus, but it doesn't just replicate its DNA. It makes RNA and then reverse transcriptase to make DNA again. So it goes through an RNA intermediate to make its DNA, which is kind of strange. Why would it bother to do that? But okay, so that's a little bit about classification. And then I just included this because it's just kind of a nice, kind of makes you appreciate how little that we know and how much there is to learn about viruses. But the different shapes, and, you know, it talks about the nucleic acid and the symmetry of the capsid, whether it's naked or enveloped, whether it's segmented or it says continuous, another way to say unsegmented, which Baltimore class it's in. So, you know, just again, just ways that we will categorize our viruses. Okay. All right, any questions, anything you want to talk about before we jump into replication? Okay, so remember, a virus has to be in a living cell before it can replicate itself. Okay, so that's kind of the difficulty in growing them up in the lab. We have to have some living cells and then, you know, add the viruses in. We can't just grow viruses on the media that we grow bacteria on. We can grow them in bacteria cells, but we can't just grow them on media. Okay, so they need a living cell in order to replicate. Usually when a virus will replicate, it's going to do some serious damage, if not kill the host cell. Now, not necessarily right away, but it's usually going to be harming the host cell. And there's 2 different kinds of replication really the second kind is just kind of a continuation of the first kind. So we have Lydic replication. We'll talk about that first. And then lysogeny is just slightly different, includes the Lydic replication cycle. Okay, so if we look at the 5 stages of Lydic replication, we could break that down into attachment, entry, synthesis, assembly, and release. All right, so let's use as an example this T4 bacteriophage. Okay, this is a double-stranded DNA virus that only infects E. coli. Okay, so where do you find bacteriophages? Anywhere you find bacteria. So they're pretty much everywhere. We consider this a virulent phage, and that just means that it will only do Lydic replication. Okay, so if we kind of blow up the first step here of that diagram, it talks first about the attachment phase. So, so if you look at the bacteriophage, it's got a capsid head, it's got this tail sheath and these tail fibers. At the end of these tail fibers, there's going to be receptors for whatever the outer membrane of that E. coli bacteria. Okay, so the bacteriophage are not moving in any kind of, they don't have a way to locomote themselves. They don't have a way to move purposely. So they are just landing on a E. coli bacteria, just random motion. Okay, so they kind of bump into it. Oh, yep, this is the kind of cell that I can infect. All right, and so it will attach again by proteins there. And then, so next, and you can see that down here, it's attaching to the outside. And then in terms of entry, it kind of looks like that it just kind of contracts and it acts like a syringe to inject its double-stranded DNA. That it's not actually that easy, because remember, even E. coli has at least a thin layer of peptidoglycan. So remember, that's pretty rigid. And usually bacteriophage needs a little help to insert its DNA through that peptidoglycan. So it will carry some lysozyme. Okay, it's a protein lysozyme. And what lysozyme does is it breaks the bonds between those sugar subunits, NAM and NAG, N-acetylmuramic acid and N-acetylglucosamine. Okay, remember that the sugars of the peptidoglycan. So it breaks those bonds, weakens that cell wall, that peptidoglycan structure. So now the nucleic acid can enter into that E. coli cell. Okay, and then the first thing, and then basically the rest of this, the capsid, the tail fibers, all that stuff just falls off. Its job is done. Okay. Once the nucleic acid is inside the cell, first thing that happens here is that some viral enzymes will break up the host chromosome. Okay, so you can actually have lots of phage infecting that cell all at once. We'll see that in a picture in a minute. Sometimes that happens. That host chromosome is degraded. And then in the synthesis phase, that just means that the virus is really taking over the host cell machinery. It's taking over the ribosomes, and it's making more of its own proteins. And it's going to take over the enzymes like the DNA, polymerase 3, and copy its own DNA. Okay, so synthesis of everything it needs to make new virions. Assembly, here's a blow up of the assembly phase. So all the things that it needs to make a new virion, they somehow assemble. There's a lot we don't understand about this process. It doesn't appear to be mediated by a whole bunch of enzymes. It appears to spontaneously come together. And again, there's a lot we don't understand about that. But they are assembled, kind of like a assembly line at GM, something like that. And then once again, with a little bit of lysozyme to weaken the cell wall, we get the release phase, okay, the lysis phase, where those virions are going to burst out of that host cell. Okay, and then they're ready to go and infect another cell. So if we look at, I love this picture here of that E. coli is in big trouble, right? It's bursting. You can see all these bacteriophage bursting out. Okay, that process, you can see here the growth curve looks a little bit different than it would if we were growing bacteria. We have, you know, one phage, say we have one phage entering that bacterial cell. It takes only about 25 minutes for all that synthesis and assembly to take place. And that's, again, in a bacteria cell. And then all at once, after about 25 minutes, we get, you know, somewhere 100 to 200 bacteriophage. That's kind of a shorthand for bacteriophage bursting out of the bacterial cell. Okay, so all at once. Okay. So the second kind of replication just kind of incorporates another little loop. Okay, so there are some bacteriophage that will infect bacteria that are able to do the lytic replication, but they can also do what we call the lysogenic replication. So our example here in this picture, we're using lambda phage, and you can see it looks similar here to the T4. It's just lacking those tail fibers. So here the proteins to attach. This is another phage that only infects E. coli. Those proteins are going to be just kind of at the end of that tail fiber. It's going to recognize the outside of E. coli. We call this a temperate bacteriophage, and that means it can do lytic and lysogenic replication. Whoops, lytic and lysogenic. My writing there. So a virulent phage can only do lytic replication, and a temperate one can do both lytic and lysogenic. And what's different, you know, the attachments is the same, entry is the same, but then what's different is that the viral DNA here is going to incorporate into the host DNA. Okay, so we call that stage the prophage. All right, so a bacteria cell that has some viral DNA incorporated into its chromosome is called a prophage. Okay. And actually, that virus can just hang out in that host chromosome for generations and generations. So that bacteria cell is dividing here and making more daughter cells, and still we have that virus into that host chromosome. So what I wondered is, well, a virus that can do both lytic and lysogenic, when would it do the lysogenic replication and when would it stick with lytic? It appears that there is some kind of regulation that happens, that a repressor gets made that would repress the virus from going into lytic replication. And at some point, usually by some environmental influence, that repressor would come off. It can come out of the lytic replication We call that induction when it's going to enter back into the Lydic replication cycle. It would, transcribe all its genes into, making all of its proteins. It would assemble and then release. Okay, so, we don't really know, how long it would stay, kind of hunkered down in that prophage state. We do know that sometimes there can be some lysogenic conversion. So that means that viral genes can cause a change in the phenotype of that host bacteria. Okay, so it makes sense. It's incorporating right there into the chromosome. And again, we call that lysogenic conversion. A couple examples of that, the bacteria that causes strep throat. Okay, most of us have had strep throat at one time or another. If that bacteria, Strep pyogenes, has been infected with a certain bacteriophage, then that organism, the bacteria, has the ability to make a certain toxin, an erythrogenic toxin that causes a bright red rash. So if you're unlucky enough to get a strain of Strep pyogenes that makes that toxin, then you can come down with scarlet fever, not just strep throat, but scarlet fever as well. We know that that's much less common today than what it used to be. Another example is the organism that causes diphtheria, Corynebacterium diphtheriae. It only makes diphtheria toxin if the cell has been infected with a lysogenic bacteriophage. Okay, and if it doesn't have, it's not able to make that toxin, then it's not going to cause disease. So we know of a few examples of that. At some point, like I said, the cell can be, the virus can kind of enter back into Lydic replication. Oftentimes there's some environmental cue, like environment is getting, not good, like the virus senses that the cell is about ready to die. So we can cause induction to happen just by shining some ultraviolet light on cells. So it could be, radiation or lack of nutrients or something like that. Or sometimes we don't really understand. It just seems to enter back into that Lydic replication. Okay. All right. So 3 kind of stages there: the prophage, the induction, and that term lysogenic conversion. Just be aware that that can happen. Okay, so I might let you go a few minutes early today, again, just because we're ahead of time, but just be aware. So we've all been talking about viruses that, enter in bacterial cells in their replication. There are a few differences if we're talking about viruses that can enter us, animals. It's the same basic replication pathways. We do have some differences because a lot of animal viruses have an envelope. So that's going to make a little bit different how the viruses will enter the cells as well as how they'll leave. They can't just burst out all at once if they're going to grab some of the host membrane. So that might usually going to need to happen a little bit differently. Remember, animal cells have a nucleus. So the virus may need to go to where the DNA is in order to replicate itself. So it might need to enter into the nucleus. And also remember, comparing to bacterial cells, what do animal cells not have the bacteria do? Cell wall, right? Okay. So because animal cells, again, lack a cell wall, that just, there's a few more differences of how the cell is the virus is gonna enter and leave. Okay, we made really good progress today. I think, like I said, we're a little ahead of schedule, so I'm gonna let you go a bit early. I do have office hours tomorrow afternoon as well as Friday morning, and I hope you have a good weekend.